In this drawing, we can see both a naked virus on the left and an enveloped virus on the right. There's also naked and enveloped viral particles on the bottom of the slide as well. Let's start in the upper left-hand corner. This is a drawing of a naked virus that has a polyhedral shape. From a distance, it might look spherical, but when you get close, you realize that it's not a true sphere. It's made out of these flattened plates, these triangular plates that all come together to create a polyhedral shape. Each one of the little yellow circles that you're looking at is a protein, and all of those proteins join together to create the capsid. A portion of the capsid has been cut away in this drawing to show us this twisted purple molecule inside, which is the genetic material. And finally, at each of the vertices of the polyhedron, there is a spike protein emerging, a naked virus. Now, compare that to the one to the right, the enveloped virus. A wedge of material has been removed in this drawing to allow us to peer inside. And you can see some very similar structure in here. You can see that purple twisted molecule. That represents the genetic material. You can see this polyhedron of these small yellow circles. This is the capsid made up of repeating protein subunits. But then there's this purplish gray layer of material surrounding the capsid. That's the envelope. And finally, we have these blue balls that represent spike proteins emerging from the surface. Remember, the envelope around an envelope virus is not of viral origin. It comes from the host cell's membrane. The spike proteins, however, are viral. They come from code found inside the viral genetic material. This is what we would refer to as a spherical shaped virus. Down at the bottom, we can see examples of helical viral particles. There's a naked helical virus on the left and an enveloped one on the right. Notice that the genetic material, again drawn in purple here, has a very different look to it than the genetic material in the polyhedral shape or the spherical shape. Notice too that the capsid is made out of those same individual protein subunits all joined together, just in an elongated helical shape. And of course, the enveloped version of this particular virus has that additional outer layer, that envelope structure. There are no spike proteins present on this drawing of the enveloped virus. This might be, for example, a plant virus that we're looking at. The capsid structure that surrounds the genetic material of viral particles is composed of repeating subunits of protein. Each one of these subunits is referred to as a capsomere. So in the drawing down on the right-hand side, the blue structures that you're looking at, each one of those would be called a capsomere. There is one protein, one particular protein found in that capsomere, and it is simply repeated over and over and over again to create the capsid structure. Now, there's good reason for having one protein creating the capsid around the genetic material, and that is, if there's only one protein, that means there only needs to be one gene to build it. The gene contains the code necessary to build that capsomere. In order for viral particles to be as efficient as possible in terms of getting in and out of host cells and getting themselves replicated, it's helpful to have a limited number of genes in their genome. This example, again, down on the right-hand side, this represents a particular type of virus called the tobacco mosaic virus. This is a virus that has been studied quite a lot because it used to be a big problem for the tobacco industry. The tobacco mosaic virus infects tobacco leaves and can spoil them before they are harvested. This virus has been researched quite a bit. We know, for example, that it contains one strand of RNA as its genetic material. That's what you're looking at in red. And it is surrounded by 2,130 individual copies of one capsomere protein. Those capsomeres are arranged side by side in this helical structure. This viral particle, because it only needs to have code to build that one particular protein, it has a very small number of genes, a very small genome, which is very easy for a host cell to replicate. So there are several different recognized shapes of viral particles. The one thing that they all have in common is that they are highly symmetrical. Viral particles just have this symmetry associated with them. So far we saw the polyhedral shape. Remember, that has triangular plates that are joined together to create the polyhedron. We've seen a spherical virus. It is a true sphere. We've seen a helical virus. Sometimes the helical viral particles are called rod-shaped particles. And finally, there's a fourth category that's referred to as complex shapes. Now, within the complex category, there are two types of viruses we should be familiar with. The virus that infects bacteria, the type of virus that infects bacteria, is called a bacteriophage. This virus has a very unique looking, very complex shape, and we'll take a look at it in just a minute. The other type of virus that has a complex shape is called a giant virus. On this slide, we're looking at those different shapes of viral particles. If we start in the upper left-hand side, we can see one of these polyhedral shapes. You can really see the triangular plates in this drawing that are all joined together to create this polyhedron. Once again, this polyhedral virus also has spike protein coming out of it. There's a spike emerging from each of the vertices where 5 plates come together. This happens to represent a type of virus called an adenovirus. You may have heard of adenoviruses before. These are one of the viral types that cause the common cold in humans. This drawing in the middle represents an influenza virus. Influenza is an animal virus. It is enveloped and it is spherical. Each one of the yellow bumps that you can see on top of the envelope represents a spike protein. Down here on the lower left, we can see again the tobacco mosaic virus drawn out for us. It has a helical shape to its capsid. Of course, all the genetic material would be inside that capsid shell. And finally, over here on the right, we have a drawing of a bacteriophage. Again, a virus that's capable of infecting bacterial cells. Notice the complex shape of this thing. It almost looks alien. It has this very unique looking head structure. It's got a neck attached to it. And then it's got these very strange looking legs, if you will. They almost look like insect legs or spider legs. And then you can see these little pointed structures right here. Those are literally used by the bacteriophage to help attach itself to the outside of bacterial cells. Very interesting particle, the bacteriophage. So, bacterial cells, just like human cells and other types of cells, they can be infected with viral particles. It's just that all of the viruses that are able to infect bacterial cells are all one type. They are all bacteriophages. Over here on the right-hand side, you're looking at an electron microscope image of a bacteriophage. It really does have this unusual, complex shape associated with it. It's got that head, it's got the neck, it's got what's called a tail sheath, it's got tail fibers, those little spidery legs. Very, very interesting looking thing, the bacteriophage. Next to the EM image, we have an artist's drawing of the bacteriophage. Now, what's interesting about a viral particle like this bacteriophage is that such a complex shape would be unlikely to come together all by itself. In other words, as the host cell does the job of transcription and translation to produce the viral proteins to build new bacteriophages, it would be unlikely that all of those proteins could come together spontaneously and create such unusual looking individual structural components. Indeed, what happens inside of bacterial cells is that certain bacterial proteins, called chaperone proteins, are going to help the viral proteins come together correctly. Like the name suggests, chaperone proteins have the job of assisting in the folding of proteins when they come together. And it's the bacteria's own chaperone proteins that are going to help all of these viral proteins come together and fold up into the correct shape to be a bacteriophage. On the lower right-hand side of the slide, we can see an electron microscope image of a giant virus. Now, giant viruses really are giant. Remember, most viral particles are somewhere between 30 and, say, 300 nanometers in diameter. A giant virus can be 1 to 1.5 micrometers in diameter. Remember, that's about the same size as a bacterial cell. Very, very interesting, very large viral particles. Giant viruses were only recently discovered in about the last 20 years. It's not that they had never been seen before. There were lots of times when these particles were found on EM images, but scientists didn't think they were viral particles because they were just too big. So many scientists thought they were just some unique type of bacteria or perhaps another type of microbe. It wasn't until very recently that scientists discovered giant viruses inside of amoeba and were able to do the work necessary to show that these are not living cells, these are just complex shaped viral particles. Giant viruses have only been found in single-celled eukaryotes. again, similar to the amoeba. They've been given very strange sounding names. Some of the giant viruses have been named Mimi virus. There's one called Mama virus and one called Mega virus. The Mimi and Mama, these are acronyms, so they stand for something else. But that's the naming system that's being used so far to describe the giant viruses. So, we know of several different shapes that viral particles come in. We know of the helical shape, the polyhedral shape, the sphere, and the complex shape. Take a look at this slide. You can see drawings of the different shapes across the top of the slide and then EM images underneath. Remember, we're talking about viral particles, so we're talking about something that is very, very small, typically somewhere between 30 and 300 nanometers in diameter. The tobacco mosaic virus, which is a helical shaped virus. The adenovirus, which is an example of a polyhedral virus. You can see the spike proteins coming off. The influenza virus is shown, which is a spherical virus. And here is a bacteriophage. This one happens to be one called bacteriophage T4. On this slide, you're looking at a drawing of the SARS-CoV-2 virus, the coronavirus that is causing the world so much grief right now. Now, like many animal viruses, the SARS-CoV-2 virus is spherical. It is an enveloped virus, again, like many animal viruses, and it has spike proteins emerging from the surface of the envelope. In this drawing, the viral particle has been cut in half so that we can peer inside. What's drawn in purple here represents the genetic material. SARS-CoV-2 happens to be a virus that uses RNA as its genetic material, and it happens to be a single strand of RNA. The protein that surrounds the genetic material doesn't look like a shell so much as a coating around the genetic material. Each one of these small purple circles represents a unit of protein, a capsomere. So again, it's not surrounding the whole piece of RNA like a shell. Instead, it's sort of laying over the RNA. That would be what we consider the capsid. Then we have the envelope, which is drawn in gray. And you can see some proteins drawn emerging from the envelope. Smaller orange and yellow proteins, and then the larger red protein. The red is the true spike protein. That's the protein that binds to receptors on our cells. We know that the SARS-CoV-2 virus binds to a receptor called an ACE2 receptor. ACE stands for angiotensin converting enzyme. That's not a term you need to learn, but it happens to be a receptor that when it binds to these spike proteins, it allows the viral particle to come into our endothelial cells, the cells that line our blood vessels. So this virus, which was first identified because it caused an unusual pneumonia in human patients, this virus is actually not a purely respiratory virus. It's a vascular virus. It binds to and gains entry to the cells that line our blood vessels, called endothelial cells. Now, the smaller proteins that you can see in the envelope, the yellow and the orange, these are referred to as envelope proteins. We are calling the yellow protein, the envelope protein E, and the orange one is being called envelope protein M. One other thing that you should know about the SARS-CoV-2 virus is that, like some viruses, it contains an enzyme within it. So a protein that is capable of performing catalysis. It acts like a catalyst when certain chemical reactions occur. Replicase is an enzyme that is not found in any other living cell. It's only found in this particular virus, at least so far. Replicase, like the name suggests, is going to do the work of helping to copy something, to replicate something. And that something is the RNA molecule inside this virus. Remember, viral particles rely on the host cell's enzymes, the host cell's ribosomes, to do the work of making copies of the virus. So the host cell has to do two things. It has to copy the genetic material of the virus, and it has to transcribe and translate the genetic material. In other words, it has to make the proteins that the genetic material codes for. This virus uses RNA as its genetic material. So the host cell will simply look at that RNA just like it looks at its own RNA, and it will treat it like messenger RNA. The host cell will simply translate the viral RNA. The ribosomes will use the RNA to make proteins, viral proteins, the proteins that make up the capsid and the envelope and spike proteins. The other problem, though, is that the viral genetic material itself needs to be copied. In other words, the host cell needs to copy the single-stranded RNA molecule so that each new viral particle that's being made inside the host cell will have its own RNA genetic material. Here's the problem, though. There is no enzyme inside of a living cell that can take an RNA molecule and use it to make more RNA. Remember the central dogma. The central dogma tells us how the cell takes its genetic material and uses it to make protein. We know that in a living cell, the DNA molecule is used to make RNA in a process called transcription, and the RNA is then used to make protein in a process called translation. There is nothing in the dogma that tells us that an RNA can be used to make another RNA. There's no enzyme to do that work. Cells can take a DNA molecule and make more DNA. That's the process that we called replication. The enzyme that does that work is called DNA polymerase. But there is no enzyme that can take an RNA and make more RNA from it. So this is a problem. Puts a big roadblock in front of this virus. So The SARS-CoV-2 virus has to have its own enzyme to do that one job that the host cell cannot do for it. The SARS-CoV-2 virus has to carry with it an enzyme that can take an RNA molecule and make more RNA from it. And that's this mysterious enzyme called replicase. Viral particles, remember, are extremely small. Viral particles cannot be seen under a light microscope. We're talking about a range of about 30 to 300 nanometers, approximately. Remember, giant viruses are the exception to that. Giant viruses can range from about 400 to about 1500 nanometers. And of course, 1500 nanometers would be the same as 1.5 micrometers. Remember, coccus-shaped bacteria tend to be about 1 micrometer across. So giant viruses are the same size as certain types of bacteria. The drawing that you can see in the upper right-hand side of this slide just reminds us of this range of size. What you're looking at over here on the left, this is a eukaryotic cell. It's got a big nucleus in it that would hold the DNA. It's got organelles floating in the cytoplasm. It's a big cell, whether it's a plant cell or an animal cell. Bacterial cells are significantly smaller than eukaryotic cells. Now, compare the bacterial cell to some of these viral particles. This one down here represents an adenovirus. Here's a spherical one that represents an influenza virus. Here's a bacteriophage. These are much smaller. We always have to remember that viruses can't be seen underneath a light microscope, so we are not going to see them in routine microbiology work.